So good evening, everybody. My name is Kelly. I'm with Lakeland Agricultural Research Association. I'm very happy to have Shane New with us from Understanding Ag. He's going to be talking a lot about nutrient management and how to get the most out of your crops and your fields for this upcoming season. Please feel free to either type your questions in the Q&A or the chat, as well if you want to raise your hand, and I can unmute you and you can lob questions during the presentation, and then we will also have a big question and answer period at the end of the session. So with that, welcome, Shane.
Thank you, Kelly, for inviting me here this evening to do a webinar for your organization. And as Kelly mentioned, if you have some questions during the presentation, feel free to raise your hand or so Kelly can unmute you, and we'll address the questions as they come in. And obviously we'll have a Q&A session after the presentation. So I'm Shane New. I'm with Understanding Ag. I'm a farmer rancher in northeastern Kansas. So if you know where Kansas City is, I'm about 90 miles northwest of Kansas City. So kind of in what would be called the tall grass prairie. Obviously, a lot of our farmland in this part of the world, our land was broken out of the prairie, farmed for a few years, and a lot of it went back to go back grass. But obviously, my passion is production agriculture. I grew up in agriculture my entire life. You know, and there's a lot of things, you know, we don't comprehend, or we're never really ever taught how things actually function. So, you know, we start off tonight's presentation, if I get my cursor to work. You know, I start off when I talk to farmers and ranchers, you know, what is farmers or ranchers? What are we in the business to do? Now, I think Kelly set up a ability for you guys to answer the question, and she has a poll here. So I'm gonna allow everybody here a minute to address the question. And usually when I give these presentations, I'm with a live audience and out in the crowd, and I allow them to respond. So I'm gonna allow you guys a minute here. And so what are we in the business to do as farmers and ranchers? Thinking on it, Kelly. Everybody's being bashful tonight. Well, I'll tell you what, I'll just answer the question here for you. So as farmers and ranchers, what are we in the business to do? Well, we're in the business to grow plants. Oh, there's our results. They're a little delayed, Kelly. So what we are in the business to do is to grow plants. So my question is to you now is, what does it take to grow a plant? I think Kelly has another question here for us. So what does it take to grow plants? We getting our answers, Kelly?
Yeah, they're coming in. I will share them with you right now.
Well, a lot of people, we need soil. You're absolutely right. We need soil to grow plants. But you know, a lot of you, you know, you capture where was the direction I was going. You know, we need some sunlight. You know, we need sunlight to grow plants. What else do we need to grow plants? Well, some of you answered that question. Obviously, we need water. But there's another thing we need to grow plants. You know, it's something we kind of forget about, but we need carbon. We need CO2. Grow plants. You know, my question is also then, if we have sunlight, we have water, and we have CO2, what do these three things all have in common? Well, the one thing they do have in common is they're all free. But you're probably thinking yourself, yeah, but we still need nitrogen, don't we? And you'd be correct. But the nice thing about nitrogen is it's also free. 78% of the Earth's atmosphere is comprised of nitrogen. It's called dinitrogen, and it's this very strong triple bond that holds this bond together. We obviously know legumes have the ability with rhizobium bacterium to fixate nitrogen, atmospheric nitrogen. But there's also a lot of free living type of soil microbiology, specifically bacteria that can break this triple bond. And they don't need an association with the plant root in order to do that. And I know you've probably heard in prior presentations, but above every acre, there's approximately about 32,000 tons of nitrogen above every acre. So my question to the group would be, how many of you are writing checks for nitrogen? How many are you starting to utilize or capture some of this? This is what we get for free. I always refer back to the Nature and Properties of Soils manual. Fertilizer, the reality is that nutrients added by normal application of fertilizers, whether organic or inorganic, incorporated into complex soil nutrient cycles. Key words here, relatively little of that fertilizer nutrient, from 10 to 60%, actually winds up in the plant being fertilized that year. So, I think of this when this is a very accurate statement. It's for every dollar you spend, probably at the very best, you're getting 50% utilization of that money that you're spending for that synthetic nutrient that's being applied. A lot of that nitrogen gets immobilized in the soil microbiology. Now, a little bit of background about myself is I'm a student of Dr. Elaine Ingham's. And one thing Dr. Elaine Ingham, talks a lot about is the soil microbiology. And this is just one eyedropper on a slide at 400 magnification. But look at all the life within the soil here. These pineapple looking structures like you see here kind of in the center of my screen, those are testate amoebas. So they're actually predators. All these little round little figures that are very, very small, those are bacteria. These darker circulations, those are fungal spores. I'm basically waiting for the proper conditions in order to germinate and start building fungal hyphae. In fact, if you can see my cursor here, there's a piece of fungal hyphae strand right there within this view. So a lot of the nutrients that we do apply gets immobilized within the soil microbiology. And you're probably thinking to yourself, what does it mean by immobilization? So immobilization is the nutrient itself is tied up within an organism, whether that be a plant structure tied up in the biomass of the biology, or even like myself, there's nutrients tied up inside of my own body. Those nutrients are now immobilized. And the only way they'll ever be remineralized again is if I perish and redistributed back through the system. This is one of these powerful videos that I always appreciate and to share. But this is a plant root sending out chemical signals out into the soil, and also organic acids. And basically it's releasing what we call root exudates. And these exudates are basically carbon, liquid sunshine. These carbons are actually polysaccharides, sugars, that the soil microbiology that I just showed you a moment ago are feeding upon. So why would a plant be willing to give up energy, sunlight, carbon, water into the soil to feed the soil microbiology? One thing we didn't really capture, at least when I was growing up in farming and agriculture, is this association between the plants and the soil microbiome. In fact, we never really acknowledge the soil microbiome. But the plant's providing root exudates, sugars, to feed the soil microbiology. But it's not a one-way street. In return, the plant's expectation is for that soil biology, this photo right here, to help dissolve nutrients from your silt, sands, and clays. So the plant could be sending out chemical signals telling the biology, I'm willing to give you some of this sunshine, some of this carbon, some of this sugar, if you can provide me maybe a little bit of sulfur here today. And the biology, basically through enzymes being released, dissolve the nutrient the plant's looking for from the silt, sand, clays within our soils. And really, at the end of the day, what we're trying to get established in any kind of a nutrient cycle, we've got to understand the ecosystem processes. We've got to understand there's got to be an energy cycle. Basically a plant capturing what we talked about earlier, sunlight, water, and carbon, along with nitrogen, providing the exudates to the soil microbiology. Not only is the soil microbiology starting to provide nutrients to the plant, it's also starting to build soil aggregation, that crumbly structure you see within your soils. That's what helps us start to achieve a water cycle. Once we start achieving a water cycle, we can start focusing then on a nutrient cycle. And we will go a little more detailed into this a little further into the presentation, but it all starts with the energy cycle. If we don't have the energy cycle functioning within our soils, we'll never get a water cycle accomplished. And if we don't have a water cycle, we'll never get a nutrient cycle. That's just the way it is. So when we go out and do consulting, which is part of what Understanding Ag's business is, we visit a lot of farms and ranches. The one thing we always do is we always look at the plants growing within the fields. And our primary focus is to look at these root structures and to see if we see any silt, sands, and clays adhered to the roots themselves. Because remember, as I showed earlier, that plant's releasing root exudates to feed the soil microbiology. A lot of times when we look at vegetation and we see no silt and sands and clays adhere to those roots, it's kind of telling us something. It's telling we probably have a lot of excess nutrients within our soils. The plant's not having to waste any energy or provide any energy as root exudates in order to feed the soil microbiology. But when we go to look at different systems, look how these roots here, obviously different type of root, more of a fibrous root, but notice how these rhizosheaths are engulfed with silt, sand, and clay particles. So basically, this is the biology feeding along this rhizosheath the root exudates that are being provided from the plant. And it all goes back to what we talked in the beginning. The sun, water, and carbon, along with some nitrogen, is helping feed the soil microbiology. One thing I'd like for you to capture on this particular image is look down on this rhizosheath where my cursor is. We're starting to see the formation of what we call the microaggregate, the small aggregate structure. So basically, as the bacteria start to feed along these rhizosheaths, they start binding your silt, sands, and clays together, start building what we call the microaggregate. And if you look on this particular image here, it's not as well defined. But if you notice right here, notice that larger aggregate structure right there where my cursor is. That's what we call a macroaggregate. That's that larger pea-size aggregation that we want to see. So it takes the bacteria to build the microaggregate, but it takes the fungal component in order to build the macroaggregate. And here's a good example of the fungal hypha within the soil that's helping weave together these larger aggregate structures. Now you're thinking to yourself, what's all this biology? Why is it really important? Think about this. Think about your root as a percent of the biomass, as a percent of the volume within a cubic foot of soil, how much of that root is touching that area of a cubic foot or cubic meter of soil? You know, it's very, very small percentage of the volume. But when we start making associations with the biology, notice how these hypha strands are engulfing throughout the entire volume of the soil structure itself. So as we apply nutrients, or nutrients are being dissolved within our soil solutions, here's your root right here. But notice how the fungal hypha is helping expand the nutrient collection, think of it that way, of that particular plant. Not only is it helping provide more nutrients back to the plant, back to the root, it's also helping start to build that macro aggregation. And here's a good example. And one fungal species we talk a lot about, it's very, very important within our soil systems, is mycorrhizal. And it's the glomalin that's produced by the mycorrhizal that helps create these larger aggregate structures, these macropores, as they sometimes are referred to. So think of it this way. Think if you have a jar of marbles, and if you pour water in the top of the jar of marbles, that water is going to dissipate itself all over probably every marble and quickly go to the bottom. That's how soils are meant to function. These macro aggregates should allow the water to infiltrate deeper into our soil profile in order so it can be held within the soil. And I know Gabe spoke to you guys last week, talking about principles. And we talk about armor on the soil, trying to keep that moisture within the soil, protecting it so it's not evaporating out from there. And the reason why moisture is not only important to grow a plant, but it's essential for the soil microbiology. The only way the soil microbiology function is is on the films of water within the soils, basically a subaquatic environment. That's how they get their motility, how they move on the films of the water. But these aggregate structures are what allows a lot of the predators, such as that testate amoeba that I showed earlier, to be able to swim to the rhizosheaths of our roots to start feeding on the bacteria that have nutrients immobilized within inside of them. And the only way to get those nutrients released back into the system is basically a predator feeding on the bacteria and fungi, taking that immobilized nutrient that's tied up in the bacteria and fungi and remineralizing that out through the predation process. So we talk a lot about mycorrhizal. So plants use 5 to 50% of the carbon from photosynthesis to feed fungi. So plants give up a lot of their root exudates, a lot of their energy back into the soil to make that relationship with the soil microbiology, not only to expand basically the nutrient pool that's being provided back to the plant, but it's also important in disease protection. Think of a lot of the diseases we deal with in agriculture. This biology is going to protect its food source. It's going to engulf these root systems and inhibit a lot of pests. Here's a good illustration of a corn plant, maize. It's engulfed with mycorrhizal, and look at a plant, the difference. But just think of the difference in the root, basically the nutrient capture that's being created with the mycorrhizal relationship, especially phosphorus and zinc. And also it's a great way of moving water back to a plant, the fungal mycorrhizal. And it's also critical in building the macroaggregation within our soils. So here's a great illustration of a corn plant with no mycorrhizal association and with mycorrhizal association. Just look at the healthiness of the roots themselves with the association with mycorrhizal. So we do a project here in Kansas with General Mills. It's called the Kansas Wheat Project. When we first started down here, we were working with a lot of different growers, and obviously they grow a lot of soybeans in that part of the world. But I want you to take a look at this rhizosheath sheath of this particular soybean plant. We don't see any silt, sands, and clays adhered to these roots at all. And if you really look, if my photo is good enough illustration, you see very little nodulation. There's a nodule right there, but you see very, very little nodulation occurring. So this is telling me something. It's telling me there's a lot of excess nutrient within the soil, especially nitrogen, that the soybean plant's not having to provide root exudates to feed the soil microbiology. So when that's occurring, it's inhibiting aggregation formation, which is going to inhibit water infiltration, which is going to also not only water filtration, but inhibit creating a good nutrient cycle. So here I'm going to show you another example. Here's another soybean plant, not too far away from where the previous one was taken, but look at the difference. Look at the aggregation, the macro aggregates that are being built along the rhizosheath of this root. Look at the nodulation that's being placed by the plant along the roots themselves too. But there's one key thing I want you to take a look at here. Maybe some of you captured it. But what do you know is really significantly different when you look at the background, especially and along the root? Between that photo and this photo, what is one thing that really highlights to you, do you think? There's a significant difference in the soil color. So this field here is very depleted in carbon. This field isn't. So there's great difference in the soil appearance. And these fields are probably less than a half mile apart. But it's tremendous difference on management. And really it goes back to how do we manage these systems and really manage for what we want. So I just love showing this photo, just an illustration of the difference between a perennial plant and an annual. So here's intermediate wheatgrass. Here's basically annual wheat. But look at the difference in the volume of fibrous root structures between the two plants. And what we're really trying to do is how do we in production agriculture mimic the natural systems? How do we mimic this? How do we make this work to our advantage? That's the reason we talk about keeping a living root in the soil as long as possible. I know in your guys' context up north there, your growing seasons are very short. And there's some challenges you guys have that are unique, but it's always following the principles of soil health. So we'll just go through this again. It's the biology that's producing the glues to help adhere the silt, sands, and clays together in order to build aggregation. It's the polysaccharides released by the bacteria that help bind particles, soil proteins, and other biochemicals that bind soil particles also. And it's also the biology of earthworms, fungal, that help physically enmesh those particles together to start building the macro and macro aggregate. And here's just a pretty cool photo that I captured when I was in Kentucky a week or two ago. But look at this earthworm. And you can notice how this earthworm's releasing basically these glues that's helping adhering the silt, sand, and clay particles together to keep its burrow open. Not only this earthworm is helping move microbiology, other bacteria, other fungal spores within the soil solution. So it's really diversity of community of the soil microbiology that helps with a lot of our, water cycle and nutrient cycles. And here's just a good example. When I was doing a lot of microscopy work, I would often see this. This is what I would always refer to. I'm not saying it's exactly correct, but you could notice how these fungal hyphae we're starting to bind things together, starting to create these structures per se within the soil solution. And at the end of the day, this is what we're trying to achieve. We're trying to achieve good soil aggregation, because we're going to have a water cycle first, energy cycle first, water cycle, then a nutrient cycle. And we're going to go a little more into the nutrient cycle here, but let's make sure we have this right. So it's the exudates being produced by the plant, capturing sunlight, water, carbon, along with nitrogen to produce exudates. That's what brings along basically the first feeders, the bacteria. The bacteria feeding along these rhizospheres, starting to hear your silt, sand, and clay particles together to start the formation of microaggregates. Next ones to come along are your fungal. The fungal help start creating the larger macroaggregate. Also, it helps increase basically the root volume within the soil, bringing nutrients further distances away back to the plant as it feeds on the root exudates itself. Then come along our predators, nematodes and protozoas. So when we have soil aggregation within our soils, these little critters here, they got to be able to swim in order to get to these roots to start feeding along these rhizospheres, where all the bacteria and fungi are that have nutrients immobilized within their bodies, within the biomass of them. And basically, these are very nutrient compact organisms. So it takes these predations to come along these rhizospheres to start feeding on the bacteria and fungi, taking those nutrients that are immobilized, remineralizing them, and making them plant available right here along the root zone where the plant needs it. So one test we like to utilize a lot is called the total nutrient digestion test. And this is a 12-inch core sample, and we encourage a lot of people to pull this test and to see what your total nutrient analysis is. But what I want to capture here is when you look at the nitrogen pool, now obviously these nutrients are bound to carbon, but look how much nitrogen was in this particular sample: 3,215 pounds, phosphorus 690, potassium 6,284 pounds. We have all the nutrients we need to grow a plant. What most soils are lacking is the biology. In order to release these nutrients, making them plant available, taking them from the organic form and converting them into the inorganic form, plant available. And it goes back to the soil microbiology. And this predation I was talking about, here's a bacterial feeding nematode right here. All this black inside his gut here, those are bacteria. And those are very nutrient dense organisms. But it takes this predation to take these bacteria that's got nutrients immobilized within their bodies, a predator come along consuming it and mineralizing those nutrients back out into the soil solution so it can be captured again by a plant. So we're going to talk just a little bit high level view of carbon and nitrogen, but basically this is immobilization and mineralization. So basically, this nematode that I just showed you a picture of, this is him right here, this Pac-Man looking little creature. You know, this nematode's carbon to nitrogen is a 30-to-1. So it takes 30 parts carbon to 1 part nitrogen to meet one metabolic cycle for this organism. So he consumes these bacteria, which have a carbon to nitrogen ratio of 5 parts carbon, 1 part nitrogen. So obviously, if we can do some simple math here, he's going to have to consume 6 of these bacteria in order to meet his carbon needs. But how much nitrogen did the nematode actually need? He only needed 1 part nitrogen. But each one of these bacteria had 1 part nitrogen. So that means he actually consumed 6 nitrogens, but needed 1. So what's he doing with the excess nitrogen? Well, he's releasing it back into the soil, mineralizing it, making it plant available, taking the immobilized nutrient that was bound up in the soil bacteria, now mineralizing it, making it now plant available. Do we have any questions, Kelly, that we need to address before I go much further? I see I have some chats or Q&As here.
Yeah, for sure. I guess number one, how do we protect our microbes or within the soil solution and the mycorrhizae that require the aqueous environment when we have been dealing with extreme heat and drought for the past couple of years?
So that's a good question. So it's really keeping those soils armored and keeping those soils covered is a huge piece of that. Whether that be through grazing activities or our cropping rotations, not bailing our residues off. That's one good way. Making sure we're spreading our residues evenly when it comes out the back end of the machine, the combine, so it's evenly dispensed on top of the soil surface. When we graze, another thing is definitely during the actively growing season is making sure we're not overgrazing, making sure if we can move those cattle, using polywire, and doing a good trample effect, keeping those soils covered. So we're holding that moisture within the soil so it's not evaporating out, and keeping that biology working and function as long as possible. Because remember, once we build an aggregate, that aggregate's life's about 21 to 28 days. Now, obviously, it's not going to be dependent upon heat, temperature, whether it be it's extreme hots or colds, how well the biology is going to function. And it's also going to be dependent upon moisture.
Okay.
Is there another question?
Yeah. So environmentally sensitive nitrogen products have been a lot more popular in the last few years. Does it provide the yield and protein advantage in, say, hard red spring wheat?
Repeat the beginning of that question again, Kelly, please.
So does environmentally sensitive nitrogen or any kind of polymerized coat nitrogen provide a yield and protein advantage in hard red spring wheat?
I'm not sure I can address that question properly. I'm assuming, are they talking about inhibitors? That's inhibiting the nitrogen from? Is that what they're talking about?
Our ESN products out here have a coating on the outside,
yes,
and so they provide like a slow release nitrogen form.
So what that's basically doing, it's inhibiting the enzymes by the microbiology from breaking down the nitrogen, is what it's doing. There's sometimes they use acids in those products that inhibit, especially urease, being, that not allowing the bacteria basically to break that down. And what are they referring to on the protein?
Sorry, does it increase protein yield or protein content?
And within the wheat, I can't answer that, to be quite honest with you. I don't know how it would. I guess I don't see the association right off the bat, how that would affect the protein content, not as the nitrogen's being held longer into the soil solution per se, that the plant's able to capture it to help with the protein component. We got to remember too, there's all kinds of forms of nitrogen that a plant can utilize. Plants can utilize amino acids within the soil solution. They can utilize proteins within the soil solution. And it's really emphasizing, there's many forms of nitrogen that a plant can capture. And I'll talk about it here just a little bit more in a second.
Okay, well, we can keep going and then see what questions come up as I go.
Thank you. So I just want to show here real quick a chemical analysis of one of those bacteria. So obviously there are a large percentage of their biomass with those little micron organisms of bacteria, 70% water, but 96% of the cell is composed of 6 elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. So they're very, very nutrient-bound organisms. And they have a lot of nutrients, as I've mentioned previously, immobilized within inside of them. So I got a cool little video here, just watching a bacterial feeding nematode feast on bacterium. But this is the true nutrient cycle. is this is how nature functioned. It's taken basically the plant providing exudates into the soil, making associations with the microbiology in return providing the plant with the nutrients that it needs using enzymes, that bacteria feeding on those exudates and the nutrients within the soil solution, immobilizing those nutrients. Then it's taken predation to come along to feed upon that microbiology in order to release the nutrients into the soil solution. Here's just another great example, and this is more of an example of the reason why diversity is so important. On the left is an image basically with bacteria and fungi only. But when you have a very diverse microbiological community, look at how the decomposition process is occurring. The big thing is we want to understand is this residues we provide on the soil surface, not only protecting the soil, but those are immobilized nutrients that the biology is able to basically capture again, remineralize it, and making it now plant available. This is our nutrient cycle. Taking immobilization, nutrients are bound up within the cell structures, the biology utilizing that for their own metabolic needs, immobilizing it, and remineralizing it back here within the soils. And so, we're really just starting to understand how the biology functions within the soil, how the nutrient cycle, really kind of does function and occurs. But there's some great research that's been done by Dr. James White. It's called rhizology, and I'm not sure how many people are aware of this, but it's really very interesting. So one thing that is known now, that plants are actually carnivores. So that may make some people a little nervous if you're a vegetarian, but really they're carnivores. And basically what Dr. James White and some team of scientists from Australia discovered was plants are actually bringing bacteria into the plant itself, through the meristem, and basically through an oxidative process. Basically think of it deholing that bacteria. And within the body of that bacteria is all those bound nutrients, immobilized. But as that bacteria, that cytoplasm moves up the plant root, basically the plant cells are poking holes in it and leaking those nutrients out of that cell. Remember I showed that graph a little bit ago, how much nutrients immobilized within that bacteria of nitrogen, oxygen, sulfur. But what's fascinating from Dr. James White's research is that basically the DNA material of the bacteria is helping as the plant releases that genetic material back outside of the plant. It's helping with root hair elongation. So it's actually helping our plant root system expand within the volume of the soil. So basically what was discovered is basically a cycle. So basically the DNA material spit back out into the soil solution. The bacteria is reassimilating itself and is basically going back a cycle. There's some research Dr. James White has released that basically from the vegetation that he studied here recently, that 30% of the nutrient needs of the plants was being captured through this rhizophagy cycle. And this is just absolutely fascinating. What's more fascinating about this, and Kelly, you mentioned to me that some of the attendees tonight are ranchers or have livestock. One thing I discovered, at least on my operation down here in Northeast Kansas, was the best seed I could produce was a seed grown on my own operation. And you're thinking to yourself, what does he mean? Well, this research kind of proved my thought process, something I knew for a while, but rhizology has also discovered that the plant is inoculating the seed with the soil microbiology that it was grown in. So what we started to discover was that we were having better results by allowing our plants to go to reproduction, then bringing our livestock in. Obviously that's a little more lignified vegetation, but allowing the animals to shatter that seed and move that seed throughout our farm and our ranch in order to help increase the basal coverage. But the reason we were having such good results was a lot of it was due to the fact that we were inoculated. The seed that was being provided was inoculated already with our soil microbiome. And here's just a great illustration. On the bottom left, here's basically bacteria feeding on the root exudates, the top, this number 2 photo. and I'm gonna use this bottom 4th photo, but you can see within inside this plant root, the bacteria themselves, they're basically having their nutrients leaked out of the plant, leaked out by the plant, then reassimilate, being spit back out through root hairs back into the soil solution. So this is absolutely incredible, fascinating research that's just starting to be discovered and acknowledged. So, really, guys, what it boils down to, what we're in the business to do is we're in the business to grow plants. And it's through the process of growing plants, providing root exudates to the soil microbiology, the biology in return provide the nutrients to the plant, also helping us create soil aggregation, which allows for better water infiltration because we need water to grow plants. That's allowing for the aggregate structures to be built and allow for the larger organisms, the predators, such as protozoas, nematodes, and other microarthropods, to come feed along those rhizosheaths, taking those nutrients that are immobilized within the soil microbiology, the bacteria and fungi, and remineralizing that right along those rhizosheaths. So those plants can capture that nutrient in order to grow biomass. So I want to we talk a little bit about tests we use to help farmers and ranchers transition, from how do we limit the amount of nutrients we need within our soil solutions. So I'm not sure how many of the attendees are familiar with the Haney soils test, but this was developed by Dr. Rick Haney. What I found absolutely fascinating with the soil test is Dr. Rick Haney said, okay, what does a plant use to help extract nutrients from the soil? And all plant roots produce organic acids, but there are 3 primary organic acids that all plant roots produce. That's oxalic, malic, and citric. So basically they take those three organic acids along with nature's solvent, which is water, and take your soil sample, take your silt, sands, and clays with those organic acids and water to capture plant available nutrients. And when we look at the fertility here, if you can see my cursor where it says fertility, that's what the H3A extract is. It's telling me how much potassium, calcium, magnesium, sodium, zinc, manganese, iron, copper, aluminum, sulfur, that's being captured with those organic acids the plant produces, along with water. Now, obviously, these are parts per million. So if you want to determine how many pounds per acre that is, you multiply that number by 2. But more importantly, when we look at a Haney soils test, like any standard soils test, we always go over here to the far left where it says other soil measures. We look at the soil pH and determine what the pH is. The next thing we like to capture is what we call the soluble salts. Now remember, anything that dissolves in water is basically a salt. But we're looking at the soluble salts within the soil solution. The next thing we like to look at is soil organic matter. So when I look at soil organic matter, I always think of organic matter as basically the home within my soil. So a 4.6% organic matter is a pretty good size home. Now it's going to depend upon your context. I know there's a lot of soils that, that are very high organic matter, and a lot of soil samples we look at are very, very low organic matters. In this particular sample, this is a good size home. But like in any home, we go down here to soil health and look where it says soil respiration. Basically, it's telling me how many occupants is what soil respiration is telling me do I have within inside that home. So a respiration at 209 parts per million CO2 is a very, very good soil respiration. So if organic matter is the size of my home, respiration is the occupants inside the home, like any home, we need a place where we store our food. And that's the way I think about organic carbon. So this organic carbon, this water extractable organic carbon, is basically the carbon that the biology is acting upon. So the exudates, the carbon that's within the soil solution, this is what the soil microbiology is feeding on, and other organic compounds of carbon. But it's like any storage refrigerator, an analogy I like to use is, it depends on what kind of food's in the refrigerator, on how well I'm going to consume it. So if my fridge at home here was filled full of Brussels sprouts, I would probably starve to death because I can't stand Brussels sprouts. But this is what microbial active carbon, MAC, is telling me. How well does the soil microbiology, the respiration here, the microbiology, how well do they like the food in the refrigerator? So 118 is they really like the food. We like to see that MAC number, microbial active carbon. We'd like to see that more around a 70, or excuse me, between a range of about a 50 to 75. Now, obviously, it's going to be a little dependent upon the time of year the sample's taken and how active the biology is, on how well and how rapidly they're consuming the carbon. The next thing we'd like to look at is carbon to nitrogen. What's my carbon to nitrogen ratio within my soil? And this is a 6-inch coil, excuse me, this is a 6-inch core sample of soil that was sent in. So my carbon to nitrogen ratio is an 8.37 to 1 ratio. So 8.37 parts carbon to 1 part nitrogen. So once we start seeing this number get below an 8, we understand that we need to get more carbon into this system. Once we start seeing these C to N ratios get above, an 18 to 1 or a 20 to 1, that's telling me we need to provide more nitrogen in the system. And that's how we go about when we look at how do we design cover crop mixes or maybe even crop rotations to help with this carbon to nitrogen ratio. Because think of it this way, think if you have a low carbon to nitrogen ratio of lower than 8 to 1, think of a fire burning. Maybe you have a campfire burning. Well, if you have a nice campfire burning, and if you have all of you have is small twigs or small branches to throw in the fire, it's going to be hard to maintain that heat. So I need bigger pieces of wood to keep that campfire where it's comfortable for everybody. So if my carbon to nitrogen ratio is below an 8 to 1, I need to provide a larger source of carbon, a bigger chunk of wood. But if my carbon to nitrogen ratio is of 20 to 1, I think of it as the campfire's basically embers, and all I have is big chunks of wood. When really what I need is those twigs and those branches to put on the fire to build that fire back up in order for I can put those larger pieces on to make a comfortable fire. So that's kind of how we go about using plants as our basically maybe our crop rotations or cover crop recommendations. The percentage of legumes to grasses, the legumes obviously provide the nitrogen. The grasses are providing a lot more carbon into the system. And how we go about balancing that. And when you see this, if you do a Haney soils test, it gives you a percentage of legumes to grasses. And the best recommendation I can utilize is whatever my normal seeding rate is of a something of like a spring wheat or a biennial, per se, let's say it's 1 million seeds per acre. What that's telling me is 20%, so that'd be about 200,000 seeds of a legume if I was using a cover crop, to about 800,000 seeds of a grass. So that's one thing you want to make sure you understand your seed counts and your typical seeding of a biennial when you look at this. The next thing the Haney takes into consideration is nitrogen. Here's a traditional nitrogen test, looking at your inorganic forms of nitrogen that was captured. The Haney takes into consideration all forms of nitrogen. And these are all plant available forms of nitrogen. The big question is that nitrogen, that nutrient, going to be available when the production crop that I'm growing is going to require its most amount of nutrient, especially nitrogen. So I take a look at this number here, 43.7. So the next thing I go look at when I'm looking at this nitrogen comparison, can I take a credit for this 43.7 pounds of nitrogen that was in this soil sample? What I'm going to go up here and look at where it says nitrogen up above here, where it says H2O extract, you'll see where it says organic nitrogen to inorganic nitrogen, a 6.32%, excuse me, 6.32 to 1. That's telling me I'm converting my organic forms of nitrogen into inorganic forms of nitrogen, making those nutrients available. So we like to see this at least above a 2.5 and probably better, closer to a 5. And it's going to be dependent upon the time of year that the soil sample's taken. Obviously, this was taken in May of last year when the soil temperatures are warming up, biological activity is occurring quickly. We're converting a lot of that organic form of nitrogen into the plant available inorganic forms of nitrate and ammonium, making them plant available. So I kind of use a triangle effect. I go down here and look at my respiration, my occupants in my house. How many occupants do I have? Do I have enough workers inside the house to help convert this organic form into inorganic? And that determines how much of this credit I will capture. So on this particular test with this kind of CO2 respiration and the conversion of organic to inorganic, I would take probably almost a full credit for this 43 pounds of nitrogen. So this is how we kind of interpret the Haney soils test. I know it's a pretty high level view pretty quickly, but I'd always be glad to work with anybody who's using the Haney soils test and help you go over and review your samples. This is another test we use real quick. It's called the phospholipid fatty acid test. I call this basically looking underneath the hood of who's down there, basically soil microbiologically. The first thing I like to take a look at is I look at the top up here where it says total biomass of the microorganisms. And I'm not too concerned about this number. I use the analogy, think of a stadium. How many attendees are within inside the stadium? How full is it? How many chairs are filled? And that's what the biomass is telling me. What I'm more interested in is how much diversity do I have within the attendees? How many are male and female? How many are wearing ball caps? How many are wearing sunglasses? I want to see more diversity. So 1.46 is a pretty good diversity number. So I've got some pretty good diversity within the soil sample. We go down here and we look at the breakdown of functional groups as a percent of the biomass within the soil sample. 47.39% of the sample is bacterially dominated. And I don't care what your sample is, you're always going to see a high percentage when a PLFA test of bacteria. But it breaks it down the bacterial community. So gram positive is 25.57. Actinomycetes, they're gram positive, but they're very identifiable, but they're 10.62. And we also have gram negative present. So our bacteria is well within check where we want to see it. The fungal is what we really are interested in. Do we have the fungal component within the soil? And as this particular sample, we have 10.63% of the biomass is fungal. But we break that down even further. We have our arbuscular mycorrhizal present at almost 3%. Our saphotrophs, who are basically our decomposers, at about 7.7%. So we do have a decent fungal presence. What I'm more interested in is, do we have a protozoa? Do we have predation occurring? In this particular sample, we do. We have a 0.86, and that's not a real high number. That's pretty good for most agricultural soils. In fact, a lot of the soil samples I look at, that's usually a 0. We have very little predation being captured in the PLFA test. And I'll just share with you real quick, the undifferentiated number, you're always going to have an undifferentiated number. We'd like to see this number as low as we can get it. I have seen it as low as 28. But most typically, it depends on how the system's being managed and a lot of other factors. It'll be around a 32 to 35. I have seen some very degraded systems up around 80% undifferentiated. And those systems are a lot of times are very anaerobic. And when you stick a tile shovel in the soil, they're very, very collapsed soil structures, no aggregation whatsoever. And you know, my buddy Ray Archuleta said it the best: the plant and soil are one. And as farmers and ranchers, that's what we're in the business to do: grow plants. We grow plants in order to produce revenue through proteins or carbohydrates. But it's the plant and the soil relationship with the soil microbiology that we're in the business to do. And no presentation is complete till you see a happy pig. So that's the end of my presentation, Kelly. If we have any questions or you want to open it up or however you want to proceed.
Sure. There is a good question here. Could you comment about having more than just the two main plant taxonomic families of the grass and the legume family in a mix in regards to soil nutrient availability and soil biological activity, such as having plants in the sunflower dandelion plant family, plantain family, brassica family, and so on and so forth?
Yeah, so a lot of those particular species, you know, we would probably put them over in the legumes just because there's, well, like brassicas, we put the brassicas over the legume component. Part of the reason why is, you know, they're basically nutrient scavengers. They draw the nutrients back up, especially nitrogen, that feed the soil microbiology. But yeah, diversity as well. The roots, the fibrous roots are what's providing a lot of carbon in the soil. But we do want to see diversity of root structures. And if we can provide diversity within the soil, that's advantageous. The big thing is we want to be conscientious, you know, really do the test in order to see where we're at with our carbon and nitrogen, where are we at with our functionality, and how do we manage the diversity of the plant species that we can grow within our environment to help address soil function.
Thank you. So much of our more conventional farming practices favor the bacterial soils. So if we're beginning our regen soil foray, how would we promote the predators and the diversity in the microflora?
I mean, yeah, first thing is understand the principles of soil health. You know, minimize that tillage. Tillage is very detrimental to soil microbiology. It basically, when we till, we diffuse a lot of oxygen in the soil, which basically wakes up what's called our strategist bacteria. They're opportunistic. They consume a lot of the carbon out of the soil structure, which dislodges the silt, sands, and clays. When we do receive moisture, those silt, sands, and clays have nothing adhering to them. So silt, sands, and clays start finding air holes, and those silt, sands, and clays plug those air holes, which inhibits water infiltration going deeper in the soil profile, which starts surface runoff of water, which is precious, which is what we need to grow plants, but also it allows for a lot of nutrient runoff to occur. So first off is making sure we're utilizing good soil health principles like no-till. How do we keep armor on that soil to keep that moisture within the soil, keep it from evaporating out? And the next thing is keep a living root in that soil, keep providing carbon into the system in order to feed the soil microbiology. Like I mentioned in the presentation, once we build an aggregate, it's not like it stays there forever. Those aggregates get consumed again by the soil microbiology, the carbon itself. So we got to be continually providing exudates during the growing season in order for aggregation to be continually provided.
So with keeping a living root and adding diversity and keeping like armoring the soil, where do you see that, like the perennial wheats, or like how do we integrate the covers into a traditional monoculture cropping situation?
Well, obviously you guys have a very short growing season, you know, And it's very difficult. Once you harvest a crop, you probably don't have enough growing days or heat units to get a cover crop planted. Depends on what rotation you have, I'm sure. You know, one thing to do, I advise a lot of clientele up north, is open the sieves up on the combine a little bit up and open the air up a little bit and maybe blow a little bit of extra seed out the back of the combine and allow that to be basically your living root, you know, until, you know, it'll be winter terminated more than likely, you know, with your conditions. But that's how I'd probably go about it within your guys' context in your environment.
Do you have thoughts on some of the like the eco teas and stuff like that that try to introduce bacteria or microbes into the soil to try and kick up the nutrient cycling?
Oh, you want to go down a rabbit hole, don't you? Um, so if you're going to use inoculants, inoculate your seed with the inoculation, because when that plant germinates, I mean, when that meristem root starts to break into the soil, it's starting to leak root exudates. So if I have the biology I'm trying to help promote, I want that to be able to one of the first feeders on those exudates, so it can help enhance build that community right there along that rhizosheath. The soil diversity of microbiology is so vast. To sit there and say we can put an inoculant out there that's going to achieve this and this and this is a little ridiculous. You know, the reason why it's important not only to get that armor on the soil, all that biology is adhered to the leaf structure and the stems. It's everywhere. That biology is blowing in the air every day. It's in your guys' snowpack. Well, if I have a leaf out here or a plant growing and those spores are blowing through the air, those leaves and stems have what's basically called body glues. And if I would take an electron microscope and I would look at a leaf structure, I would see a lot of that same microbiology on that leaf that I see in the soil. Now, obviously, when a plant goes to maturity, it's no longer feeding the biology also along that leaf and stem. But those body glues are keeping that biology adhered to it. So the biology it basically sporulates up, goes to sleep. But if I can get that down on my soil surface, and as those body glues break down, you know, that biology is going to get released again. It may blow from here to Timbuktu, but there's also a thing called gravity. And if I have that residue on my soil surface, guess what? That biology now can enter into my soil solution and help promote more diversity within the system. So it's also, we got to think about this. This goes back to carbon and nitrogen. You know, think of a legume plant as a very low carbon and nitrogen. So bacteria can break that down very easily. So that's going to help build up bacterial communities. But if I'm trying to promote more fungal, you know, I need a higher carbon than nitrogen, because the only thing that's going to break down that higher carbon, that a more lignified material, is going to be fungal. So if I'm trying to promote per se, I got to provide what the biology is seeking or needs in order to help propagate it. So that's another way you can do it. If you are going to introduce biologicals into your system, I encourage, one more time, encourage the check strips. Apply it and leave an area unapplied. And then compare, did it really benefit you for the dollars invested?
Excellent answer. So to talk about more carbon-based, like what is your thoughts on the humic products? So things like Humulate, Black Earth, like there's many different humic and fulvic products out there. What are your thoughts on those?
I don't, a lot of people are putting them in in with their nitrogen sources. It's helping hold that nitrogen in the soil, obviously carbon. So I don't see a whole lot wrong with using that. It's like anything else, leave check strips if you can, and see if the product that you're purchasing is really benefiting you. And I'll be all honest with you, really, it's really adhering to those 6 principles of soil health. Understand your context, minimize that disruption. I don't care if it's grazing or row cropping, keep that soil covered. And then obviously a living root, provide diversity. Diversity is huge. One of the biggest challenges in my part of the world, we used to have a lot of diversity down here, but obviously commodity prices, the direction of a lot of different things, it's turned into corn and soybeans. And it's probably been decades now, almost 2 decades, since wheat's been grown down here. Well, adding a new crop within your system is a new disruption. It's new diversity. It's new root exudates being fed that soil microbiology. So it's usually going to have a very positive impact. And the best analogy I can use is think about if you maybe you don't exercise very often, but you decide you're going to go train for a marathon. You wouldn't go out and just run 1 mile a day. You would do different disruptions to train your body, your muscles, endurance, resiliency, to be able to compete in a marathon. Well, we want to do the same type of process to our soils. We want to provide different disruptions and different diversity of plants. That's how we build resiliency, build diversity of soil microbiology also.
So in times of drought, soil bacteria will protect themselves with a waxy coating. If the conditions are severe enough, this waxy coating can cause hydrophobic soil surface conditions, preventing water infiltration. How do we overcome this waxy coating? The obvious answer for them is that to have soil armor in place to prevent the coating from forming. But if our soils are currently hydrophobic, what can we do to get over this?
They'll get over it themselves with hydration. So yeah, biology, it's pretty fascinating. So bacteria have a unique ability to produce what's called saturated and unsaturated fatty acids. It's a survival mechanism. They can produce in very, very drought conditions that waxy coating. And you'll see times you can put water on top of a soil surface and it basically peds up and doesn't infiltrate. Usually we see that in a lot of times definitely in the southwestern part of the United States on some of these soils. It really goes back to, it's going to take hydration, but it's making sure we have principles in place, in order to, once we do get blessed with that hydration, we can start changing the functionality of what we're seeing occur. Animal impact has a huge role in that. I've worked with some ranchers in Mexico where those crusty soils, they're using the animals basically walking them through those areas in long, narrow, skinny paddocks. Now, obviously, sometimes they're having to bale graze or provide a carbon source to basically jumpstart. But it's the manure, urine, the hoof action that's breaking some of that crusting, and the manure and urine, bringing biology in that's helping provide conditions in order to grow a plant. And when they do get blessed with some moisture, helping some of that moisture start to infiltrate.
Will having more residue on the soil surface solve issues like anaerobic soils?
Oh yeah, it's going to help. Obviously, keeping that moisture in the soil as long as possible will keep the biology functioning, but it's really going to be, do we have enough carbon within the soil, to feed bacteria? So that's a piece of it. That's a very big piece of it, keeping that soil covered. But it's also making sure we have enough carbon. Because the bacteria aren't like you and I. I mean, what I refer to like that, if you like, you see behind me here, my office, you know, I have no other food source inside this office. The bacteria can consume that drywall. They can consume, they're going to eat the house, is what I'm trying to say. So once they run out of carbon, they're going to start going after aggregate structure. Once they're out of aggregate structure, they're going to start consuming organic matter. So basically, they're going to keep finding something to consume carbon to feed a pond, which can have an unintended consequence, which is going to destroy water infiltration. So the armor is very important. It's making sure we got enough carbon being supplied to the soil microbiology for them to keep consuming.
We talk lots about nitrogen, but we don't focus very much on phosphorus. So what conditions are most economical to apply phosphorus? Or how do we improve our phosphorus cycling?
Well, it's a biological process like nitrogen also. Mycorrhizal is a huge piece of that. Mycorrhizal loves to go find phosphorus. The types of phosphorus utilization, I know some producers using rock phosphate as a phosphorus source. Obviously, it's going to take a little longer for utilization. But one of the big things is really go back and pull that Haney soils test. On that test that I showed, it does show plant available phos. And capture and see how much phosphorus you do have up. In all honesty, phosphorus doesn't move through the soil profile very, very quickly. Basically, it moves about 1/4 of an inch per season. So if you applied phosphorus in the past, you probably have a decent amount of phosphorus within your soils. Sometimes what we see is there's such an excess of phosphorus, the plant's not having to send out the chemical signals to make the association with mycorrhizal, provide the phosphorus source. So really, you're actually inhibiting a natural occurrence of a mycorrhizal relationship with the plant and the biology by having too much phosphorus. Because why would the plant give up any energy to provide the chemical signals to make an association if there's plenty of phosphorus within the solution of the root that it doesn't need it?
I'm not sure if you want to touch a little bit on like micronutrients. Like in our area, we're very short, like copper, selenium, that kind of stuff. And I'm just wondering if you have thoughts on either how to get it a little bit, like make it more available, or just adding it in. I guess, what are your thoughts on micronutrients?
If you're deficient and you're seeing deficiencies within the crops themselves, obviously, you're probably gonna have to supplement. One good thing to do is do the TND, total nutrient digestion. See if you're really lacking those nutrients, or are you lacking the biology to help provide the nutrient to the plant. So there's a few things I would really encourage anyone to do baseline testing. Do the total nutrient digestion. That's a 12-inch core sample. I would look at doing some Haney soils testing also. When we do a Haney soils test, let's hypothetically say we have a 160-acre field. What I would do is split that field in half. I'd pick 4 or 5 locations within that field, one half of it, and I would put those samples 6 inches deep in a 5-gallon bucket, 4 samples at each location, mix them together, and send it off as one composite sample. As long as the field's been farmed the same, same crop species and everything, same nutrient applications, it's going to be a pretty decent analysis of what's going on within that soil. So that's what I would encourage to do. The big thing on the Haney soils test, and obviously, we like to see the soil temperature at least 55 degrees Fahrenheit. Now, obviously, most of your guys are going to be planting up in that part of the world, and the soil temperatures are that warm. But really, on nutrient management, I would really encourage split applications too of your nutrients. you're gonna get far greater utilization of the nutrient that's being applied versus trying to put it all down at one time. Because a lot of that nutrient you're applying at one time is going to get immobilized in the biology and not be utilized by the plant, or it's going to be, lost through leaching or back into the atmosphere as nitrous oxide or maybe even dinitrogen.
Are plants the only organisms that produce organic acids, or can fungi produce organic acids like oxalic?
Fungi can produce organic acids. I'm not specific which organic acids fungi exactly produce. You make me go back to my microbiology books. I'd be more glad to. But so one thing I do want to share, obviously we deal with a lot of compaction in agricultural soils, even grassland, rangeland situations and areas. One thing I find truly amazing is, the best plant roots we have can break about 300 pounds compaction. Those fungal hypha can break 1,100 pounds compaction. So I mean, obviously, the reason we really want that biology functioning, it can help break a lot of compacted areas within our soils. Just another important role they play.
So looking back over your regenerative journey, is there one thing that you wish you did sooner, or what was the biggest important change that you made in your operation?
Good question. Really being intentional. And what I mean by being intentional, I've known Gabe Brown for a long time. And when I first heard Gabe Brown speak, I thought, I'm already doing what you're talking about. We're in a 36-inch rainfall environment where I can get away with a lot of stuff because we were blessed with moisture. And one thing I used to do is I used to remove a lot of biomass in the form of hay and silage, feed cattle. And it finally caught up with me one year. I removed all my biomass. And what I would do is I would remove that biomass, usually rye or triticale in the spring, and I'd come back in and no-till soybeans in. And it worked really well for several years. And the one year I did it, and I got caught. And what I mean by got caught, it quit raining. And I had no armor on my soil. My soils turned as hard as concrete. I had basically lost about $100,000 in production. And that was a very shocking moment for me. That really illustrated to me, you got to be intentional. You got to keep armor on these soils. And from that point on, I never removed silage ever again. Now we still do a little bit of haying, but we've minimized our haying down to, in fact, I'm selling my big round baler here in 3 weeks on a sale. It's really being intentional and being adherent to those principles. That's the only way you're ever going to get soil to function. And it's very rewarding once you do get soils to function, what they really can do. And another thing, I'll throw it out here just for food for thought. We really got to ask ourselves a really important question. Are we trying to outproduce our environment? And what I mean by that is, I'm going to use an example. I'm going to pick on my own home state here, Kansas. In western Kansas, we can grow 300 bushel corn in a 10, 12-inch rainfall environment because they have the ability to irrigate. The unfortunate problem is we've almost pumped all the irrigation water out of the Ogallala aquifer. Just because we can doesn't mean we should. And what I'm trying to emphasize is sometimes we're trying to outproduce what our environment's going to actually provide. And what's the unintended consequences once we run out of water? And in those environments, there's going to be no more production.
Where do you see the regenerative agriculture movement going in the future?
Well, I really do feel, I feel that it is the future. I feel it's resilience in our systems. Obviously, I'm not going to get into this subject matter, but there's a lot of things occurring within the world: supply chains, distribution, demographics. One thing I see a major challenge coming about is, having enough employees, distribution. So what I'm referring to is, if I'm where I've got to use, I'm dependent upon synthetics or whatever it may be for my system to be functional. And if there's a supply chain issue with either labor or distribution or whatever it may be, it doesn't bother me anymore. We're not dependent no longer on that. It's built a tremendous amount of resilience in my system, and it's helped make my operation profitable. And really, it's helping me, and I hate to admit this, but I'm getting to the age, where I've got to start thinking about succession. I want something that my kids want to come back to. And, if it's a system that's very dependent and very uneconomical, not profitable, then why would they want to come back to it? Why would they want to be a part of it? And so that's part of my context, my why, but it's also being driven by consumers. I mean, we talk to a vast amount of supply chains that are scrambling, trying to figure out, because the consumer demand is, we want to make an association with the production who's producing our food. And we also want something that's very nutrient dense. And a lot of the nutrient density comes from the association with the plant and soil microbiology, getting these systems back to function again. I'm not saying we're ever going to get away from synthetics, but how do we manage them better and better utilization? And so we're not so dependent upon it.
So you mentioned that Elaine Ingham played a significant role in your microbiology education. Was there any other people that played a role in your journey?
Dr. Lanningham played a huge impact. I mean, obviously it opened me up to the world. I'm a pretty enthusiastic reader. I never was. That was a new disruption in my life, forced myself to read. Jeff Lowenfeld has got some great books out, Teaming with Microbes, Teaming with Fungi. Hold on a second, I'll pull a book here. Michael Phillips wrote a very, very great book called Mycorrhizal Planet. Very good read. Really goes into the importance of the mycorrhiza within our soils, and also the soil microbiology. But there's a lot of good books out there. Dr. Chris Nichols, Dr. Christine Jones. Obviously, I've spent time with those individuals and read a lot of their research and their information. Great resources. Walter Yenny is another good one from Australia.
Perfect. Well, I think we gave everybody lots of food for thought and some additional reading that they can follow up with. So seeing no other questions, I want to thank you very much for presenting tonight and sharing your information. It's been wonderful to hear from you and also meet you for the first time. And thank everybody else for joining us for this evening, so we can wrap it up here.
Well, Kelly, I appreciate the opportunity and always looking forward to getting up into Canada and seeing your guys' beautiful part of the world and getting to meet some of these producers up there.
For sure. Well, thank you very much and enjoy the rest of your night.
Thank you, Kelly. Thank you, everyone.